Thank you, and I have to apologise. There's some issue with my camera, so you're not going to be able to see me, but hopefully you can hear me. Are you able to hear me okay?
Yes, we can hear you, yeah.
Okay, yes, apologies. I dropped my camera and it just will not work. So I'm just going to share my screen now. Hopefully you can see that. Oh, is that no? Hold on.
I have your presentation here if you want me to open it, Pamela.
No, it's okay. Sorry, I'll share the second screen. So I was just sharing the wrong screen. So you should be able to see that now. Is that okay?
Yeah.
Okay, great. So thank you for inviting me. My talk is going to be more research based. I'm going to tell you about some of the things that we're doing here at Queen's to create greener solutions in biotechnology and to look for a more sustainable algal-based bioeconomy. So we know that microalgae are photosynthetic microorganisms, just like seaweeds and things. They transform light energy into chemical energy that produce compounds. So the compounds produced on the cell wall depends on the species. For example, you can get red microalgae that produces things like anthoxanthin. So our earlier speaker spoke about that in crabs. Whereas diatoms producing compounds like fucoxanthins. These species also have different types of cellular walls. Like diatoms, they have inorganic cell walls from silica. So it's these inorganic cell walls that we're interested in as potential medical applications. So Queen's University is based in Belfast, but we do have a marine lab, which is based in Portaferry. So it's just around the peninsula, and it's located here. I've added a photograph of our marine lab, so we have all sorts of facilities based there. And I've got some set images of some of the culturing work that we do. We have small scale and larger scale facilities. We also go out ourselves and collect samples. This is a photograph of one of my collaborators who came over from the US, Professor Matt Julius, to help us collect diatom samples for some of our medical research. And to ID the samples that we collect. We also would get cultures from SAMS in Scotland from culture banks, or we can get them from him as well. Our facilities have micro and macro hatcheries. We also have wave tanks for different types of experiments, and we have sites that can also harvest seaweed on long lines out in Strangford Loch, and sites for tidal wave energy research. So that's a little bit about our facilities. So the species that we're interested in, and I'm going to present some on, are diatoms. And as I said, we're interested in the inorganic cell wall of these. And we're also interested in them because not only do they have this inorganic cell wall, but they also have proteins and fatty acids. So if we can find an alternative use for the waste product, so the byproduct, if you scale these up, that's an added commercial value to these products themselves. So silica in bone health. So as I said, we look for medical applications. So my research focuses on orthopedics. And we're interested in seeing if these greener or if this silica from the diatoms can be used in orthopedic applications. So the link was discovered in 1972. Taking silica supplements orally was found to increase bone health. Since then, silica has been incorporated into biomaterials. There's an example of a product, BioGlass, which was produced by Larry Hench in Imperial College London. And it's like a filler material that has been produced from synthetic silica. But the issue with that material is that it uses very toxic chemicals to produce, and it's also produced at high temperatures. So that has an issue in terms of the green credentials of that product. It does work well, but there is issues around producing the compounds themselves. Diatom silica offers a natural alternative that grows under ambient temperature and conditions. So growing the microalgae is like a little factory that basically produces this silica that we're interested in for potential orthopedics. Now, every strain of diatoms produce different types of cell wall. So this is one of the types. We've looked at different, and we've tested them to see how suitable they are for bone repair. So this is just some SEM, scanning electron microscopy micrographs. So you can see the inorganic structure. So the organics has been removed from these samples. And you can see the particle size and the surface topography of the filler material. And then at the bottom, we have some AFM images. We can't, however, add the silica particles directly into the body. We need some type of system to carry them in. So we've looked at adding polymer gels we're incorporating them into polymer gels. So this is one study that we've done. We did this out with Northwestern University, and we've created these silica gels to incorporate in our silica particles. So the one on the top is the gels only, and the one on the bottom has our diatom silica added into it. So we've added two types of polymers together. We've then functionalized the surface of the diatoms with an amine group or a thiol group to basically create a stronger bond between the polymer fraction and the silica. We've done lots of different tests in these in terms of mechanical testing and biological testing, and I've given you an example of some of the mechanical testing here. So here in this graph, you can see when we have our gels only, we have our modulus of between 100 to 150. And as we increase the concentration of silica, the modulus increases, which is advantageous in bone repair because your bones are loaded. So something with better modulus is better. When we get to higher concentrations, we can see that the modulus starts to drop down again. So there is a sweet spot between 50 and 100 milligrams per milliliter when adding in the silica particles. We've looked at not functionalizing this, so just adding the diatoms directly to the gels, but we find that if we add this additional linker, which is basically a way of binding it to the gels, we get better modulus. So the darker one is without, and the lighter one is with a linker. So you can see that we are getting better mechanical properties. So that's one way that we've looked at adding it in to a system which you can directly implant. So you can see here the tweezers, you can easily lift up the gels and they can potentially be used as an implant material. The silica itself also provides a means to attract the cells. So it attracts bone cells. It's osteogenic, which is an advantage too in adding it instead of just adding the gels on their own. So that's one example of how we've added them in. Another way that we've looked at adding them in is using FDM, so 3D printing. So we basically create here, we use extrusion to create a polymer. So this is PDLGA, which is a medical grade polymer, and we blend the diatom samples in. So we produce filaments that have both polymer and silica in them. This is then 3D printed into a 3D scaffold that can then be implanted directly into the body. So again, with this, we have had some good results in terms of the mechanical properties and the preliminary cell studies on that work. So both of these studies we're moving forward to look at it in animal models to see how it performs when it's implanted. So now I'm going to move on to something slightly different. So this is looking at the sustainability side of the seaweed. So this is completely different work. It's a new direction. And our last speaker spoke a little bit about bioremediation. She mentioned it in terms of seaweeds, whereas this we're looking at kind of the bioremediation process in terms of the microalgae. So this is basically creating like a closed loop system where nutrients from the farm are basically fed back into the microalgae and used as a fertilizer. Just like to point out that we're not using this type of algae as a biomaterial. That would be a long way off in terms of regulations and all of it. But we're just saying if we can actually grow the algae on the organic fertilizer that's available here in Northern Ireland. So different AD producers will feed their AD plants with different feedstocks, and that results in different nutrient profiles. So one solution may not work for all. So we're looking at screening different fertilizers to grow the effluent or to grow the algae. Apologies. So this is one of the examples or one of the studies that we've done, and it's in collaboration with Agri-AD. So this is one of the suppliers, AD suppliers that we've worked with. And we've looked at culturing Pterodactylum, which is a marine diatom. And this, we looked at all different concentrations, but we actually find it grew quite well in just 1% digestate, because the digestate is really rich in organic or in the nutrients required to grow the cultures. So this is some of the data that we've got from adding just 1% as opposed to using the F2 media. So this is an example of our cultures. Now they're very dark in color, and that's not because of the effluent that we've basically put into the cultures. It's the strain of microalgae that we're using. So it's the diatom microalgae, as I said, Pterodactylum. So when it grows up and gets dense, the cultures change to this color. So it's not from the effluent itself. It hasn't discolored the media. The larger culture here is an example of a different kind of green algae that we've grown on the effluent nutrients. So that's some of the work that we're looking at. This experiment here, so this is the experiment that I'm presenting the results on. We're just using a standard lighting because we're basically seeing if the organic fertilizer will work to grow the nutrients. When we characterize the organic fertilizer, so the AD effluent, we find that it was deficient in phosphate, which was necessary for the microalgae to grow. And certainly it was cited in the literature that it is required. So we added nitrogen to both our control and our organic fertilizer to boost the growth, and we quantified the fatty acid and amino acid content of the cultures. So here is some of the results that we've got. So this is just our total fatty acid profiles that we've got, and we're comparing it. The 3 on the left hand side is grown in commercial media, so F2. So we have the F2 media only, and then we have the F2 media supplemented with a different concentration of phosphate to it. And then on the right hand side, we have our organic media only. So this has been grown without any phosphate added, and then with, again, the same concentrations of phosphate added. And this is grown with 1% effluent only. So in this one, that's using the organic AD media, we haven't added any of our commercial media, so it's just been supplemented into the seawater. So I should point out that adding the effluent is not a matter of just adding it directly from the AD suppliers. We do put it through a number of processes and filtrations, activated carbon to remove some of the matter that we don't want and to isolate the nutrients that we are interested in. So you can see that we do get similar profiles if we compare the F2 media without any phosphate in it, which is not what we were expecting, directly to the microalgal culture. You can see statistically they're relatively similar. When we added the phosphate to both of our cultures, or to both of our conditions, the F2 only and the organic fertilizer, we got a reduction in the total fatty acids. So it actually had a negative effect, which we weren't expecting. We then looked at the overall profile, the individual profile of fatty acids. So it's not a matter of just producing bulk fatty acids, but are those fatty acids of interest? And apologies, I think I've highlighted the wrong one here in this square, but the difference in terms of the C16 fatty acids, we find between the F2 and the organic fertilizer was different. We saw a reduction in these fatty acids in the organic fertilizer. And then highlighted in this one, we can see that in both of the medias produced EPA, so an omega-3 fatty acid, which is desirable, and we got around the same concentration in both. The other thing that we also profiled was the amino acid content, and we find no difference in the amino acids apart from one of the amino acids, which I've highlighted here, between the medias themselves. So overall, I've just highlighted some of the issues and barriers that needs to be overcome if we're considering using AD effluent and actually microalgae in general. There are currently less than 10 strains of microalgae that's been approved for commercial use beyond fish food. You tend to find that there's certain species that are used all the time commercially. So there is a need to kind of push for more strains to come out. There's also the issue, and some speakers or some attendees have been asking about this, the economic costs, and it's certainly something, cost viability of producing microalgae, and it's certainly something that industry are very interested in. Some of the things that we came up against, so we worked with the AD sector, but we also worked with some of the feed industries to see about how we bring these solutions forward and can we actually produce feeds and things. Some of the issues which you're probably all well aware of is the regulations. So there needs to be new regulations, both in terms of the regulations of the AD effluent itself, but then looking at it when it is used as a nutrient for growing something. So at the moment, the effluent itself comes under the same regulations as just general slurry. There needs to be better regulations also in terms of what is fed into the tanks, and they need to be placed with the AD suppliers. More testing is required in terms of transition of pathogens, viruses, that is whenever you're growing up these cultures, and regulations and safety in terms of the nutrients from the effluent waste, if we're going to potentially use these and have solutions for food, feed and medical products. There's also the consumer perception, which can basically make or break a product. If people think they're eating something that is produced from agricultural waste, they're not going to eat that. Whereas in reality is you're actually doing a series of processes to remove pathogens, to remove all of the nasty things out of the effluent, and you're only actually taking out the nutrients to feed back into the microalgae themselves, but it's the customer perception around that. So that's just really a brief overview on some of the research that we're doing here at Queen's. Like to thank some of my PhD students who are involved in this work, and some of the colleagues that I worked with on this, and also our funders who helped fund this research, both industry and RC UK funding. And here is some of my contact details. So thank you.
Okay, thank you very much, Pamela, for that overview of your work. Currently, I'm just going to go to the questions there. I can't see any questions, Cliona. I'm not sure if you're getting any, but to kick off the questions, I guess people are shy. I'll start with one. So in relation to the species that you grew on the waste streams there, Pamela, what are the intended applications for the amino acids and the fatty acids there? Or was it, is there a use for them currently beyond fertilizer?
Yes, well, that's one of the things we were looking at. We were looking at the potential of using them as a feed ingredient, but at this stage for animal feeds, at this stage we were only looking at the feasibility of growing them. A larger piece would need to go around the regulations and making sure that we address the questions from the feed sector in terms of transfer of pathogens and those type of areas. There are certainly other applications, microalgae. You can grow different strains, can produce biopolymers and things, and you can use them for non-medical applications. So there's a whole host of different applications depending on basically what species you grow and what type of application you want.
But can you, because I work in this area a little myself, can you use algae growing on waste products for medical applications, like say the silicone application that you mentioned at the start? No.
Well, not at the minute because of the regulations, but that goes the same in terms of, you know, everything kind of in the economy sector.
Yes.
There's issues in terms of regulations for a lot of different things that you're doing. So, you know, there is a shift, and certainly in Europe, there's a shift to look at using different agricultural wastes or byproducts and streams, relabeling them, looking at regulations of how they can actually be integrated into different food systems and things. I mean, at the end of the day, we do need to create more circular environments, and the food sector and different sectors, different industry are producing all of these nutrients, which are basically being wasted. So, you know, the one that we looked at, and I mean, there's different research in this area, we're just simply isolating like the nutrients of interest. You know, you go through different filtration steps that you remove things from the effluent waste. So it's the nutrients that you're using, but you would have to go and look at the regulations. Certainly in terms of the chemical profiles that we've seen, we haven't grown the cultures yet. We're looking at that to produce the silica cell walls that we're interested in, but certainly in terms of the amino acids and fatty acids, the profiles are very similar. There's no real difference. So, you know, you are replacing fossil-based fertilizer with an organic fertilizer to grow them.
Okay, and just following up on that then, Rodrigo has a question. Can you use them as animal feed? Which I guess the answer is
no. At the moment, as I said, there's a lot of regulations that you can go through, but certainly for the animal feed industry, it is of interest because you're getting the same types of profiles. So, you know, how do you move on and look at the regulations around that to see if you can actually get them into the medical field, or sorry, not the medical field, the animal food sector.
Yeah.
And also, you know, is there other waste streams, maybe not effluent, that you're getting nutrients that could potentially use?
Okay, and then just to follow up, Rodrigo has said to reduce enteric methane production in cattle. Sure, that's still a feed, I guess.
I mean, that is still feed, but I mean, yeah, you're not getting the compounds, I guess, that are of interest for methane reduction are the ones produced in seaweeds. You're looking at the red seaweeds. So it's nothing really that you would get from these microalgae or the brown seaweeds, you know, the polyphenol content. So no, they wouldn't be for methane reduction. There's no microalgae that I'm aware of that have compounds that are potentially methane inhibitors. So
I think there is some work on fatty acids in respect of that, but I could be wrong. It's very preliminary.
I think it's probably pretty much preliminary,
yeah. So just in relation to the silicon aspect, I guess I just have a quick question about the species that you used there. Is that a marine species or a freshwater? And I suppose, what is the percentage content in that species? If you know the answer of silicon, or you know what amount, maybe you covered it and maybe I missed it, but I just
no, we didn't. So cyclotella, you can get both. There's both freshwater and marine micro, or is it cyclotella that you can grow. So we've grown up a strain from the US that was sent over by our partners at St. Cloud that has a very similar morphology to the marine cyclotella. So the profiles are very similar, and the type of silica is the same. The actual percentage that you get, it depends on how long you keep them in culture. So it ranges between 20 to 30%. So the longer you culture them, the thicker the cell wall.
Okay, so that's a lot, isn't it?
Somebody Aspergopsis, that's a seaweed.
Yeah, sorry, I'm not sure, Rodrigo, if I'll just allow you to talk maybe there. What institute are you from? No? Okay. Okay, so yeah, I can see no other questions in the comment box there. So thanks again, Pamela, for your very interesting presentation. And there's two more, but maybe we'll address them online in the interest of time. If you can type answers, Pamela, that would be great. And Cliona, I'll hand over to yourself to close out this session.
Certainly. I just want to say a huge thank you to our speakers today, and of course to Maria and Mara, who did a fantastic job organising today's event. I wanted to mention the next subsequent event in the series, which is happening 2 weeks Thursday, so 2 weeks from today, on the 10th of the 11th, again from 2 p.m. And we have Maria herself presenting on the utilisation of fisheries byproducts for food, feed and pet food applications. We will have Dr. Ronan Cooney from BIM talking about circular economy and marine product utilisation, and Sarah Daverin, I suppose, giving an overview of the Interreg and Northwest Europe funding opportunities for SMEs. We'll have, I suppose, a link to be sent to you for registration. Some of you have already registered for today's event. It's automatically registered for the next one. And we'll have a third and fourth subsequent event, which will be circulated to you shortly. So gur míle maithagaí asach Teagasc Nua. Thank you so much for coming today. I hope you found the speakers very insightful. I'm sure I did anyways. So gur míle maithagaí agus feicimid sib ag an caiiciis. We'll see you in 2 weeks. John.